Good evening, everyone. Thank you for joining us for our From the Hill to Your Home event. It's great to see so many familiar faces tonight and to reconnect with CMB alumni from across the US and Canada. My name is Adnan Alrubaye, and I know many of you guys on this call. I'm a fellow UFA alum, having received my master's degree in science education and PhD in cell and molecular biology program from the University of Arkansas in 2013. I now have the great pleasure of serving as a research assistant professor in poultry science and biological sciences and the associate director of cell and molecular biology program here at the UFA. Before we begin, I would like for you to update your screens by adding your name and the screen you graduated, so everyone can know which, your name and the year you graduated, please. Also, please make sure to mute your microphones. You are welcome to send messages to one another or to the full group using the Zoom chat feature. I'll give you a minute to update your screens with your name and the year you graduated, if you're a graduate. Some of you guys are still with us, you don't need to do anything, just your name. It's good to see you, Dennis. Long time no see. All right, now it's my distinct pleasure to introduce three very special guests, Dean Pat Koski, Dean Jim Gigantino, and Dean Kurt Rome. Dr. Koski was appointed as Dean of the Graduate School and International Education last November. And previously, she served as the Associate Dean of the Graduate School for 22 years. Yes, let's give Dean Koski a round of applause to our new Dean. Long time overdue. Anyway, so Dean Koski is a passionate advocate for the graduate students, graduate interdisciplinary program education, and CMB program. Again, she served as the Associate Dean for 22 years. Dr. Gigantino was appointed as Associate Dean for the Graduate School in January. Welcome, Dean Gigantino, aboard. Most recently served as the Chair of the Department of History and previously as Associate Chair and Graduate Coordinator. Welcome, Deans Koski, Dean Gigantino, and Dean Kurt Rome. Dean Koski, would you like to make a few opening remarks? I yield the floor to you, as our politicians like to say.
All right. Thank you, Adnan, so much. And hello, everyone. It's really good to see you. Thank you all for being here. So this past year has been kind of a year, hasn't it? And if I had to just identify a few highlights from GSIE, it would be the following. First of all, of course, the hiring of Dr. Gigantino as the new Associate Dean for the Graduate School is kind of fun, replacing myself and watching him take on the job. He's doing a great job. And with gratitude to our amazing alumni and friends and a lot of hard work by Julie Olson, just since the day we went remote last year on March 20th, 2020, we were able to raise over $58,000 in emergency funds, which we awarded to more than 218 students, domestic and international. I'm telling you, the need last year was so great, and the students were so grateful, allowed them, many of them, to stay in school when they would not have been able to otherwise. And then, of course, all year long, we pivoted from in-person services to remote services, back to in-person services. We dealt with change a lot, and I think we did a good job. We're still dealing with change. We continued our longtime traditions, just in a different format. So we had two in-person commencement ceremonies, one in December and one in May, with social distancing and masks. We had all of our usual award ceremonies, mostly virtual, but some face-to-face with social distancing and masks. We continued to reach out to our students to see how they were doing and to make sure they were okay. And we created a graduate student mental health advisory committee because mental well-being is on everyone's mind right now. As alumni, and welcome alumni, thank you for being here. As alumni and students of cell and molecular biology, you all know the value of an interdisciplinary degree program. And CEMB is a very good and very large degree program, thanks to the efforts of Dr. Rhodes and Dr. Alrubaye. But you should know that the university agrees with you on the value of interdisciplinary work. With a generous gift from the Walton Family Foundation, the university has embarked on a venture called I-R, the Institute for Integrative and Innovative Research, which is all about interdisciplinary research, collaboration, and study. Cell and molecular biology faculty will be a very important part of this effort. The university is currently searching for the director of the institute and the first few faculty hires. So it's been another busy year for GSIE with a few unforeseen twists, but we've made it through and are looking forward to seeing you next fall at our alumni events, which you will hear more about shortly. It is, after all, the 150th birthday of our university. So again, thank you for being with us. Thank you for being our alumni and students, and thank you for staying engaged. Adnan, back to you.
Thank you, Dean Koski. I really appreciate your remarks. Now, I'd like also to thank Julie Olson, our Director of Development and Alumni Relations for the Graduate School and International Education, for joining us this evening. Julie, if you can wave at us, please, so everyone knows where Julie is. Welcome, Julie. And now it's my distinct pleasure to introduce Dr. Rhodes, our guest speaker for tonight. Dr. Rhodes, the floor is yours.
I'm going to share my screen. Whoop. Whoop. Hang on. Hang on, folks. I'm having a issue with Zoom.
It's a good representation for what we've been going through this entire year. Everything is good until it's not.
That's sort of the word for the year, isn't it? I'm having trouble with Zoom.
Yep. Or you're zoomed out.
Or you're muted. You're still muted.
I see your lips moving, but I don't hear you.
Only lately, I've not only had my microphone muted, my camera's been muted.
Okay. Can people see my share?
Yes, we can.
Yes.
All right. So I wanted to give a little history and the progress of the CEMB program. And it's still not cooperating very much. So for those that don't know, the CEMB program was conceived in 1994 in part to create a new environment at the university for interdisciplinary research in the areas of cell biology and molecular biology. It took us really until 2000 to get that through all the paperwork at the state level and get approval. We started with the master's degree in 2000 and the PhD in 2001. The original director was my good friend and colleague, John Kirby, who served for 6 years before he left and is now the dean at Rhode Island University. And I became the director in 2006 and have continued to serve in that capacity until the present. We were very fortunate through negotiations with the provost and then with the graduate school and the College of Arts and Sciences to bring on Dr. Alrubaye as the associate director in 2013 and Dr. Alrubaye was instrumental with some very creative ideas in how to grow the program, and I'll show that in a few minutes. And then in 2018, the graduate school was kind enough to give us half of the funding for an administrative support and student application specialist, Julie Brogan, who has been instrumental in helping us with recruiting brochures, forms, and working with applicants. Julie is shared between the graduate and international recruiting and admissions office and CEMB. And she's been great in helping us keep up with paperwork, accounting, and working with students in all sorts of aspects. I'm happy to report that CEMB has become one of the 2 largest PhD programs at the University of Arkansas and has been battling with electrical engineering for who is tops in PhD. And you can see that the enrollment for MS and PhD in the top right. And then we've been very successful in graduating a number of students, and that's in the bottom right for the degrees by year. We currently have 96 students that are enrolled, but we have 22 admissions offers that are out for the fall of 2021. One of the things I'm most proud about in our program is that we have been working very diligently to recruit students from around the world, and we currently have 8 current Fulbright Fellows in the program, and we have offers to 3 new Fulbright Fellows for the fall. One of the things that Dr. Alrubaye and I have worked very hard on is increasing our representation from international students, and we currently sit at 70% international students from an amazing 28 different countries around the world. And we're also 62% female. So we are very happy with our demographics in the program. The current numbers for the program, we have 117 faculty in 16 different departments. Our latest reports from 2020, in terms of grant support, supporting CEMB students, continuing grants total 28 million in 2020, and new grants almost 9 million. And in 2020, there were 34 publications with CEMB students as co-authors. So I think the program is being very successful. You can see for some of the 16 departments, the breakdown of where those students are located, and we have very good participation across the campus. Dr. Alrubaye and I have been working to increase the enrollment and increase the diversity. We've been on recruiting trips to India, Kazakhstan, the ones you see on the left. And we are hoping once the pandemic is over to have additional recruiting trips to Indonesia, Japan, Panama, and Egypt. We were interrupted in our trip to Egypt and Indonesia because of the pandemic. I cannot stress enough that the graduate school has been very supportive of this and has provided some significant funding, with the exception of during the pandemic, for these recruiting efforts. And so the graduate school has been working very hard to increase the enrollment in all of the interdisciplinary programs. And CEMB has been very happy with the support we've had. Now, we have some of the existing students that are going to be presenting tonight, and what I've selected some students that represent the diversity of this program. And for the first group of students, I'm going to ask them to give a brief explanation of what they're working on, and we're going to take them in order. Our first student will be Peter Gan, who came here as a Fulbright Fellow from Laog City, Philippines, and he's working with Dr. Srivastava in crop soil environmental science. So Peter, if you can unmute yourself and give us a few minutes about what you're working on, please.
Thank you so much, Dr. Rhodes, for that introduction. Good evening, everyone. I am Peter James Gan from the Philippines, as Dr. Rhodes have mentioned. I came to the University of Arkansas in 2018 as a Fulbright Scholar. I'm currently working with Dr. Srivastava at the Crop Soils and Environmental Sciences Department. Our interest in our lab is gene regulation through different promoters and gene editing techniques such as CRISPR-Cas9. So we apply these techniques in plants to understand biological processes that can be used to improve growth, reproduction, yield, and product quality. In our lab, I started understanding the dynamics of the starch biosynthetic genes and how their coordination affects granular morphology. So recently, we have elucidated genotype and environment-dependent mechanisms causing improper packing of starch that leads to poor grain quality. A paper on this one, which is I am primarily authoring, has been accepted for publication just the other day. Currently, I am looking into the role of two hydrogen ion translocating genes. We have generated like CRISPR-Cas9 lines on these genes, and we are looking at how their suppression affects the consumption of ATP, disturbing cytosolic pH, and inhibiting enzymes in starch biosynthesis to understand their role in grain quality, and lastly, breakdown to comprehend their function in germination. So that's just a background of what we do in the lab. And thank you for listening, and thank you for having me here, Dr. Rhodes.
Thank you, Peter. I would add that that's in rice.
Oh, yeah, it's in rice. I forgot to mention it's in rice.
Our next presenter is Celeste Dunn, who's from Rogers, Arkansas, and she's working with Dr. Kartik Balachandran in biomedical engineering. Celeste, you're on.
Thank you, Dr. Rhodes. It's good to be here today. I was actually raised in Phoenix, Arizona, but I love Rogers so much that I'll claim it. So I'll be from Rogers today. I did my undergrad at Brigham Young University, and I came here about 4 years ago. I'm a fourth-year PhD student, hoping to graduate soon. I'm in the biomedical engineering department, which I really love working with engineers. We touched on, it's an interdisciplinary program, and I very much feel that. My focus of my research is on traumatic brain injuries. The first half of my research was really on an in vivo mouse model, but the last half of my research has been on developing and validating an in vitro blood-brain barrier on chip model using induced pluripotent stem cells. So I'm excited to be here and to meet all of you, and thank you for having me.
Thank you, Celeste. Our next speaker is Layla Methib, who came here on a Saudi Arabian cultural mission scholarship and is working with somebody named Dr. Douglas Rhodes in biological sciences. So Layla.
Thank you very much, Dr. Rhodes, and hello, everybody. My name is Layla Methib, and I have a scholarship, like what Dr. Rhodes says, from Saudi Arabia. I did my bachelor's and master's degree in King Saudi University, Riyadh, of course, in Saudi Arabia. And I got this scholarship in order to complete the my PhD in the United States. Basically, I work in ascites project, which are disease caused by accumulating fluid in the abdominal cavity in a broiler chicken. I'm focusing in this project on the mitochondrial biogenesis and the relationship between mitochondrial enzymes and ascites. This study is very important to study this disease. I mean, this project is very important to study this disease because it costs a lot of money every year, the poultry industry, and waste many chickens each year. Thank you very much. And it's my pleasure to be in this meeting.
All right, thank you, Layla. Our next presenter will be Vivek Kumar, who came from Chennai, India. I have been to Chennai, it's a lovely town. And he's working with Dr. Maradi in chemistry, biochemistry, And when he's not working in the lab, he's out chasing birds in Northwest Arkansas because it's a great place to go bird watching. So Vivek.
Thank you, Dr. Rhodes. Hi, everyone. My name is Vivek Kumar. I joined the U of A in 2017, and I'm a fourth-year PhD student in the Moradi lab in the Department of Chemistry and Biochemistry. My research is fully computational and focuses on the molecular dynamic simulations of proteins. Currently, I'm investigating the structural and conformational dynamics of large glycoproteins that you might be familiar with, influenza hemagglutinin, and the spike protein of SARS coronaviruses 1 and 2. And I'm particularly interested in large-scale conformational changes that contribute to the activation or inactivation of these proteins. I'll give you a brief intro to my spike protein project. It's been demonstrated by several studies that the receptor binding domain of the SARS spike protein binds to the human ACE2 receptor. Several experimental studies have focused on binding affinity assays, but no one has really looked at the conformational changes that drive the transition of the receptor binding domain from an inactive down state to an active up state. And we're trying to see how this process differs between the spike proteins for SARS-CoV-1 and SARS-CoV-2. Thank you.
Thank you, Vivek. Our next presenter is Made Harumi Padmaswari, who came here as on a Fulbright scholarship from Gianyar, Bali, Indonesia. She's the first Indonesian student in the CEMB program, and she's working with Dr. Chris Nelson in chemical engineering.
Thank you, Dr. Rhodes. Please let me correct that Dr. Nelson is from biomedical engineering, so it's BMEG.
Sorry.
Okay. So hello everyone, I'm Harumi. I joined University of Arkansas in 2019, and I just graduated from CEMB master program this May. Then I continue to join CEMB again for my PhD in this fall. So I'm originally from Bali, Indonesia, and I work with Dr. Nelson in genetics engineering lab. My work is focusing to improve the safety of CRISPR-Cas delivery system because there are still many challenges in delivering the CRISPR-Cas component. In our lab, we are developing a safe herbicide model to ensure the safety and also the efficiency of transgene delivery specific for muscular diseases. And also we develop lipid nanoparticle as a non-viral delivery. The main goal of my research is to provide a new alternative for muscular diseases therapy. Thank you for having me.
Thank you, Madi. Next, we're going to go to Indotimi Apulu, who's from Port Harcourt, Nigeria, and he's now working. He started with Dr. Fred Spiegel, but he's now working with Dr. Nakanishi in biological sciences. Timmy.
Hello, everyone. Yes, thank you for that introduction. My name is Ndosme Apulu, but as Dr. Rose said, I am called Timmy. I initially started with Dr. Fred Spiegel studying the comparative genomics of amoebozoa, but I transitioned to Dr. Nakanishi's lab at the start of last year. So, at the start of the pandemic and everything. Dr. Nakanishi's lab focuses on using comparative molecular genetics to look for the development of the nervous system across animals. How I fit into that is I'm particularly interested in the developmental origin and evolution of particular nerve cell types. The nerve cell types are mechanosensory cells, which include your hair cells, like for us, which are in our inner ear, and other chemosensory cell types. My research is to use some of these reverse genetics techniques and cell imaging and cell tracing and embryology techniques to figure out what the progenitors of some of these hair cell types are and to figure out what the gene regulatory network are. A quick snapshot of what I've been doing is I have been able to use some cheap-seq analysis to figure out targets of some genes which are terminal selectors for nerve cell types, which allow nerve cells to be mature and develop properly. I've been able to find targets of some of these things in our model organisms, the sea anemone, and I am currently making mutants and doing cell imaging to see if I can find any progenitor cell types and get a better idea of what the gene regulatory network for sea anemone is. That's what I currently do, and thank you, everyone.
Thank you, Timmy. I'm going to mention Ronalda Proco, who will be talking a little bit in a few minutes. But she came to the program from Tirana, Albania, and she's working with Dr. Egan in entomology and plant pathology. But she's not going to talk just yet because Dr. Alrubaye will be introducing her. We chose Ronalda to give a presentation because she's one of the recent winners of the 3-minute thesis competition, which the CEMB program has competed heavily in. I would mention that Peter Gann, who spoke first, is also a winner of the 3-minute thesis competition. So with that, I'm going to stop sharing and turn it back to Dr. Alrubaye.
Thank you, Dr. Rhodes. It gives me pleasure to introduce Ms. Ronalda Proco, who is a current doctoral student in the cell and molecular biology program. And as Dr. Rhodes mentioned, she just won recently the 3-minute competition for the campus. Ronalda, please go ahead and share your presentation about your research.
Thank you, Dr. Alrubaye. Thank you, Dr. Rhodes. And CEMB, it's a wonderful family. So I'm so excited to be here tonight. Can you see my screen?
Yes, we can.
Okay. Do you remember the first days when the pandemic hit the country? We ran to the grocery stores to grab groceries, and one of the items that we grabbed was rice. Well, rice is a staple food for the half population of the world. And one of the main challenges that he's facing is a rice blast disease, which is causing 10 till 30% of a global rice harvest annually. This devastating disease is caused by a filamentous fungi called Magnoportha oryzae, which is not affecting only rice, but also is affecting wheat. It's affecting our daily bread. This fungi has an amazing organism. Once it lays on the rice plant, rice leaves, it creates a dome-shaped structure called appressoria. Inside of appressoria, this fungi generate a huge pressure as it is like equal as 40 times as a car tire. So this pressure, this fungi used to get into the leaves of the rice. But what is the most important is that in the base of this dome-shaped structure that I mentioned, there are some proteins called septins, which stay together as pieces of Lego to form a ring, which direct the force into the rice leaves. So by creating this ring, this fungi is important to cause this disease, and it caused a huge loss that feeding 60 million people. So our goal is to understand how these proteins stay together in time and in space in order to provide more tools to fight this devastating disease and provide a global food for everyone and a good quality food for everyone in the world. So this is.
All right. Thank you, Ronaldo. We really appreciate you taking the time to share your research with us. And now I'd like to open it for questions. I want to also say before we move forward that we're so proud of our current CEMB students and CEMB alumni. It really makes us happy to see you guys succeed. And achieve your academic and life career goals. So I'll open for questions. If you guys have any questions to Dean Koski, Dean Gigantino, Dean Rome, Dr. Rhodes, or myself, please unmute and ask us questions, or you can type them into the chat. Well, did we answer all of your questions? Looks like. Any questions?
I have a question.
Go ahead, Timmy.
So, thank you, Ronaldo, for your talk. I guess this is not to try to go off tangent, but I'm guessing, so part of your research is, stopping this fungus from attacking rice in some way, the rice leaves, right? So, I was gonna ask, what part particularly are you guys targeting? Is it the Apesorium itself, or maybe the septin, I can't speak English, the septin proteins that stop the Apesorium from binding to the leaves? So that's the main focus of what you're targeting, or are you like looking at something very radical, like stopping, sort of reducing the pressure in the fungi or something?
Thank you for your question. So our focus is mostly on Apresoria, so the septins that are localized in Apresoria, as we think that these septins, once they form in the base of Apresoria, they are crucial for the infection. So we suspect that this is very important for establishing the disease. And of course, these septins are also in the fungi during the development, but our focus is mostly on the Apresoria.
So currently, your research has been able to show more or less what you gave us a very, very quick, nice synopsis, that the septins are what is causing that binding. Yeah. So the next stage is like therapies, or like some form of way to stop the interaction, right? Is that the hope?
Well, our focus is to investigate which are these, like how these septins interact with each other, but also how it interact with the other proteins, because they are not alone, so they interact with the others. So maybe these will having this list of proteins, maybe this will be essential for designing new fungicide. But not from us, because we don't design the fungicides.
I see.
I thank you, Tim, and thank you, Arnaldo. Any more questions?
I just wanted to know, like during COVID, last year, was there, significant reduction in admissions from international students in the CEMB program, or how was it compared to other years?
I think we should bring in Dr. Dean Rome to speak about how the pandemic affected international admissions in general, if he's still around. I could speak to CEMB. So I'll go ahead with CEMB, and then if Dr. Rome wants to say anything. We had a large number of admissions offers for CEMB for fall of 2020, but many of those students just couldn't get here. They couldn't leave their country, and so they've been dribbling in, and unfortunately COVID also interrupted the awarding of scholarships to some of the CEMB students. So we saw a little bit of a drop in total enrollment for fall of 2020. I would add, though, that the students that were in the program made very good progress on their degree programs, on their research. It was a very trying time. The laboratories, for the most part, were shut down for almost 5 months before we could really get things going again. And it did create a few hardships. The graduate school was great in helping with funding, bridge funding. We've under Dean Koski's leadership, there's been a number of new programs to help cover these sorts of hardships. The only problem that we had were there were a few scholarship programs which, with no latitude, terminated the scholarships, but those students are finishing, and so the university dealt with this really well at the graduate level in handling the disruption. And so I'm looking forward to, now that the pandemic seems to be subsiding, we're looking for a big boost in enrollment for the fall.
And Dr. Rhodes, if I could talk about
how would I ever stop you?
Well, you've never been able to, so I don't think you should try.
You outrank me at every level.
And out-talk you, so. But I did pull up our enrollment report. In fall 2020, we had 700 international students who were enrolled, and that was compared to 745 in fall 2019. So it could have been worse. We could have had a more difficult time than that. It will be interesting to see what happens in fall 2021. Now, one of the things, of course, that was going on in fall 2020 and summer is that students, international students who were here, did not have an opportunity, didn't have a way to get home. And so a lot of them stayed because they couldn't leave the country, which is why our emergency funds were so important, because they didn't have jobs, they didn't have assistantships, they would normally have gone home and had jobs there. And so our emergency funds, and again, thank you to Julie Olson for all of your work on this, really made it possible for us to retain these students. But we didn't have as big a drop at the graduate level. We had a pretty big drop at the undergraduate level. We went from, at the undergraduate level in fall 2019, we had 662 students, went down to 509 in fall 2020. So it was worse there. Graduate, I'm hopeful. I hope we'll start picking up again for this next fall. Dr. Gigantino, do you want to add anything to that?
Yeah, the only thing I'll add is, we're seeing some really strong interest in our graduate programs from international applicants for the fall. The deciding factor, I think, for what's going to happen in the fall is that question of, can the students get to Arkansas? Number one, with COVID restrictions in some places in the world, and then even before that, can they get visa appointments at embassies and consulates abroad? I was just dealing with a student in the College of Education and Health Professions that really desperately wants to come, and unfortunately cannot get a visa appointment until February. And so she's trying, and our students have been very innovative and very adamant about trying to come. They'll go to third countries to try to get visa appointments at embassies and consulates outside of their home country. So I think that's one of our big limiting factors, especially I was dealing with a different student. You know, they are coming from India, and the embassies and consulates are closed, and so we have a lot of you know, interest. It's the question of, can we get the students here? So we're hopeful that as things continue to turn around, that we can improve that, and then once they're here, we're in a really, I think, good position in Arkansas as we move forward for the fall. So we're hopeful, but it's not over yet, but we're very hopeful for the future.
Thank you, Dean Giagantino.
If I could add, for the recruiting side of things, one of the things that we are really looking forward to is that Dr. Alrubaye and I went to Riyadh, Saudi Arabia, a little more than about a year and a half ago, and we are very pleased to announce that the Saudi Arabian Cultural Mission has given us special status to add more fully sponsored students from Saudi Arabia. We were under a little bit of a ceiling, and they were severely limiting our ability to recruit additional students. Our program, the university, is very popular with the Saudi students, and we have a number of new student applicants in the works, and SACEMB gave us the latitude to have many more students than they allow any other university to have, because of our interdisciplinary program and the fact that we have so many faculty that are involved in it. I would add that Dr. Peter Unger from anthropology has been in concert with Dean Rome, has been working on recruiting more students from Indonesia. Madi presented, and we have several new students that are also coming on Indonesian scholarships. So we are really hoping that we can really expand our recruiting in some of these other countries to help increase the diversity and the whole world aspect of the CEMB program, because we are very proud of being an international program. I could add, and perhaps Dr. Alrubaye can speak to this, that Iraq is coming back online now, and so we're having new applicants from Iraq, and we're working in Iran now, and there's a lot of interest from Iran in coming to the University of Arkansas, because we have a very good reputation in Fayetteville for being a welcoming community.
Thank you, Dean Koski, Dean Rome, and Dean G, and Dr. Rhodes for helping answer this question. I'd like to ask if you guys have any final questions before we close the meeting for today. I also would like to add that